Hello, everyone, and welcome to today's webinar, Combating AMR and Bacterial Infections and Investigating the Immune Response. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by PerkinElmer. To learn more, visit perkinelmer.com. Now, we encourage you to participate today by submitting any questions you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of our presentation. And you may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd like to now welcome our speakers. Nathan Archer, Assistant Professor in the Department of Dermatology, Johns Hopkins School of Medicine, and Stephen Baker, Professor of Molecular Microbiology at the University of Cambridge. Welcome to both of you. Nathan, you may now begin your presentation.
Hello, everyone. My name is Nathan Archer, and I'm an Assistant Professor in the Department of Dermatology at the Johns Hopkins School of Medicine. Today, we'll be talking about our research in infections, inflammation, and immunity, and how we're using this to develop better therapeutics in the skin and beyond. We'll break our talk into 3 different parts today. The first, talking about infection models and how we can use them to test different therapeutics. Additionally, better understanding the immune responses of host defense and therapies. And lastly, better understanding relevant disease models of atopic dermatitis and psoriasis, and better understanding the immune mechanisms that drive inflammation in these diseases. Our lab is primarily focused on Staphylococcus aureus, and the reason we're interested in this is because Staph aureus is the primary cause of skin and soft tissue infections in humans. It has various clinical manifestations, such as impetigo, folliculitis, and cellulitis. Additionally, Staph aureus skin infections can lead to invasive and life-threatening infections such as orthopedic implant infections and sepsis. Antibiotic resistance has been rising ever since the onset of antibiotics such as penicillin. Additionally, a recent study in Lancet found that greater than 50% of clinical isolates are now methicillin-resistant Staph aureus strains. Moreover, Staphylococcus aureus is now the second leading cause of deaths associated with antibiotic-resistant infections, highlighting the importance of understanding the immune responses against this skin pathogen. Given that antibiotic resistance is outpacing the development of novel antibiotics, and all current vaccine trials have failed to date, including the study by Merck, which found that vaccinated individuals had higher mortality rates than unvaccinated individuals, this highlights the need for the development of alternative treatment options. Our lab is using different strategies for the development of novel therapeutics against Staph aureus skin infections, including the use of new antibiotics, as in partnership with the TB Alliance, the use of new vaccines, as we are working with integrated biotherapeutics on a multi-component toxoid vaccine, and lastly, better understanding the immune responses that are protective against Staph aureus infections for the development of host-directed immune-based therapies. Our lab uses different Staph aureus infection models, including skin infection models and orthopedic implant-associated infection models, where we use a bioluminescent strain of community-associated methicillin-resistant Staph aureus and infect B6 mice and track these mice over time using an in vivo imaging system developed by PerkinElmer to quantify the bacterial burden over time in the same animal. And as you can see in the bottom right, this is a representative image of a skin infection where you can see the bioluminescent bacteria in the skin. In this first study, we were interested in understanding the efficacy of a novel antibiotic called TBI-223, which is in the same family as linezolid. And we were interested in testing TBI-223 because it's reported to have reduced cytotoxicity compared to linezolid, which is typically used for treatment of Staph aureus infections. And what we did is we did a head-to-head study comparing efficacy of TBI-223 to linezolid in a Staph aureus skin infection model. And as you can see using the bioluminescent strain of Staph aureus, that both antibiotics had reduced bacterial burden compared to sham-treated controls, and that they had similar efficacy between TBI and linezolid-treated animals, suggesting that TBI 223 is in fact efficacious against Staph aureus skin infections. And to confirm these results, we performed CFU counts and found that a similar pattern emerged, where there was reduced significance in the bacterial burdens in antibiotic-treated animals compared to sham control. In this next study, our collaborators at Johns Hopkins used PET scans in patients with infected bone to see whether rifampin antibiotic could penetrate effectively into the bone, if this could explain why infections in bone are difficult to treat with antibiotics. And what they found is over time that there's a significant amount of antibiotic buildup in the liver and initial buildup in the plasma that's reduced over time. But interestingly enough, there's a significant reduction in the amount of antibiotics that are found in the infected and uninfected bone, suggesting that a reason that antibiotics may not be very effective in bone infections is the low penetration into the bone tissue. Additionally, they found that if you increase the rifampin concentration in an animal model, that you get a significant increase in the penetration of rifampin concentrations into the bone, suggesting that increasing the concentration of antibiotics may be a more effective means of treating antibiotic infections than the current doses given. Given this information, we are interested in testing whether we could shorten the standard care of antibiotic course with a higher dose of rifampin. To do this, we used a mouse model of orthopedic implant-associated infection, where we gave one group vancomycin for 6 weeks, another group standard rifampin with vancomycin for 6 weeks, and another group with high-dose rifampin with vancomycin for 4 weeks. What we found is that both vancomycin and rifampin combination therapies have significant reductions in bacterial burden compared to vancomycin alone. This suggested that increasing the dose of rifampin allowed us to shorten the course of antibiotics in a standard care model. Another exciting treatment modality is the use of antibiotic-coated implants. In this study, we performed orthopedic implant-associated infections in rabbits. The rabbits were given implants either coated with antibiotics or without antibiotics. And as you can see, rabbits that were given the linezolid rifampin combination therapy on the implant had significant reductions in bacterial burden than those that were not coated with antibiotics, suggesting that this is an exciting new treatment for orthopedic implant-associated infections. Our lab is also interested in studying the host defense responses against Staph aureus skin infections. Upon a Staph aureus skin infection, there's an initial recruitment of innate immune cells such as dendritic cells, macrophages, and neutrophils to the site of the infection. These innate immune cells release IL-1 alpha, IL-1 beta, IL-6, and IL-23. These innate cytokines promote the development of IL-17 responses from gamma delta T cells and Th17 cells. IL-17 acts on keratinocytes to promote the production of cytokines, chemokines that help to promote neutrophil recruitment and neutrophilic abscess formation that's important for the containment and eventual clearance of the Staph aureus skin infection. To help us better understand the immune responses against Staph aureus skin infections, we use fluorescent reporter strains of mice that have either TD tomato, GFP, or citrine under the promoters of IL-17A, IL-17F, and ASC. This allows us to visualize the expression of these cytokines and inflammasome components upon the Staph aureus skin infection, allows us to quantify the expression, and understand the kinetics of when these cytokines and innate immune components are expressed. And when in combination with a bioluminescent strain of Staph aureus, we can correlate the bacterial burden with the cytokine response. For example, in this study, we were interested in understanding whether T-cell migration was important for host defense against Staph aureus skin infections. To model this, we used the IL-17AF reporter mice and treated them with FTY, which is a compound that prevents the migration of T cells from the draining lymph node into the skin, or a vehicle control. And what you can see here is by bioluminescence, there's a significant increase in the bacterial burden in mice treated with FTY, suggesting that T-cell migration is in fact important for host defense. However, to take this a step further, we looked at whether this affected the IL-17 expression in the skin. And in fact, we do see a significant reduction in IL-17 expression in mice that were treated with FTY compared to control. We saw a similar pattern with IL-17F, where we see a reduction in mice treated with FTY. This suggested that not only is migration of T cells into the skin important, but this also is important for the IL-17 expression, which is important for host defense in the skin. In this next study, we collaborated with Integrated Biotherapeutics, which has developed a novel multi-component toxoid vaccine against Staph aureus. And what we found is that vaccinated animals had significant reductions in bacterial burden compared to unvaccinated controls. However, we wanted to understand what were the immune mechanisms of protection mediated by this vaccine. To do this, we used the IL-17A/F reporter mouse and found that in vaccinated animals, there was a significant increase in the IL-17A expression in the skin upon vaccination and prior to infection compared to naive animals that were unvaccinated or uninfected. This was rather exciting and suggests that the vaccine promotes IL-17 producing T cell influx into the skin. And our next steps are to identify what these T cells are and are they important for mediating protection for this vaccine. In this next study, we were interested in understanding the role of the inflammasome in host defense against Staph aureus skin infections. As the inflammasome is important for IL-1 beta activation and release, and we know from previous studies that IL-1 beta is crucial for host defense against Staph aureus. To understand this, we used an ASC citrine reporter mouse. ASC is an important adapter molecule for formation of the inflammasome. And as you can see here, 6 hours after infection, we see a significant increase in the expression of ASC, which is indicative of inflammasome formation in the skin. However, when we used a pan-caspase inhibitor called QVD-OPH, we see a significant reduction in the inflammasome formation in the skin. However, whether this was relevant for host defense or not, we looked at bacterial burden using bioluminescence again. And surprisingly, what we found is that the use of the pan-caspase inhibitor actually promoted host defense rather than inhibiting host defense as we originally thought. This was really exciting as it suggested that pan-caspase inhibition could act as a novel therapeutic strategy against Staph aureus skin infections. In fact, when tested against Pseudomonas aeruginosa and Streptococcus pyogenes, we found a similar effect, suggesting that this inhibition by pan-caspase inhibitors was an important broad-spectrum antimicrobial therapy. Next, I'll talk about our research into understanding the immune mechanisms that drive inflammatory skin disorders, such as atopic dermatitis. Atopic dermatitis is an inflammatory, relapsing, non-contagious itchy skin disorder that affects up to 20% of children. Most children will outgrow atopic dermatitis. However, 3 to 5% of the adult population will have persistent issues. There is a high financial burden and serious psychosocial effects to this disease. The reason we're interested in understanding atopic dermatitis is that Staphylococcus aureus almost nearly universally colonize the skin of atopic dermatitis patients. In addition, Staph aureus correlates with disease severity, and highly toxigenic strains tend to predominate on the lesional skin of patients. Furthermore, IgE levels are increased in atopic dermatitis, which is an important antibody isotype in allergic diseases, and it's been found that Staph aureus promotes the production of IgE in these patients. So, the questions we wanted to answer are, what are the immune mechanisms of Staph aureus-mediated AD skin inflammation? One immune cell type we were interested in studying were eosinophils. Eosinophils have various roles in the body. They've been shown to have host protective roles, pro-inflammatory roles, and immunoregulatory roles. They've also been shown to be important for protection against helminth infections. And lastly, eosinophils are shown to have increased abundance in the skin of atopic dermatitis patients. However, it's unknown what role they have in promoting skin inflammation. Therefore, we set out to determine what effect eosinophils had in atopic dermatitis. To do this, we've used an established skin inflammation model where mice are shaved and nared, Staph aureus is applied to the skin, wrapped in bandages, for 7 days. After 7 days, the bandages are removed, and we see an area of skin inflammation that mimics AD in human patients. The first thing we did was compared wild-type mice to eosinophil-deficient mice. And what we found in this model is that eosinophil-deficient mice had significantly reduced skin inflammation compared to wild-type controls, as measured by disease score. Additionally, when we looked at the skin by H&E, we found that there's a significant reduction in the epidermal thickness in the eosinophil-deficient mice compared to wild-type controls, suggesting that, in fact, eosinophils are important for promoting skin inflammation. In previous studies, we found that IL-17 was crucial for the skin inflammation. In addition, in RNA-seq data, we found that eosinophils were associated with the expression of IL-17 in the skin. To determine what cells were producing IL-17, we again used our IL-17A/F reporter mice. And what we found is that when we apply Staph aureus to the skin, we see a significant increase in both the IL-17A and IL-17F expression as measured by fluorescence. What we did next was took the cells out of the skin and performed flow cytometry for IL-17A and IL-17F positive cells using the TD tomato and GFP fluorescence. What we found surprisingly was that eosinophils were a major producer of IL-17A and IL-17F in the skin, suggesting that the IL-17 derived from eosinophils was important for skin inflammation. To determine the human relevance of these findings, we took skin biopsies from atopic dermatitis patients and healthy control patients and looked at IL-17A and eosinophils in the skin. As you can see in healthy control patients, there's low amounts of IL-17A and eosinophils as measured by EPX expression, which is a major eosinophil protein. And when we zoom in on this, we see that there's separation between IL-17 expression and eosinophils. However, when we look at atopic dermatitis skin, we see an abundance of IL-17A and abundance of eosinophils in the skin. And when we zoom in and look at the merged image, we see by yellow and white that there's an overlap between the IL-17A and eosinophils in the skin of atopic dermatitis patients, suggesting that in human AD patients, there is eosinophils producing IL-17. Although we found the host defense responses that were important for driving skin inflammation in our atopic dermatitis model, we want to understand what role did Staph aureus play in driving this skin inflammation. And we got a hint from a previous report by the Richard Gallo group that found that Staph aureus proteases were important for driving skin inflammation. So we acquired an extracellular protease deficient strain of Staph aureus and found that when we used this strain compared to the parent wild type strain, there was a significant reduction in skin inflammation in our model as well. This also corresponded to a significant decrease in the epidermal thickening compared to the parent strain, suggesting that Staph aureus proteases play a role in promoting skin inflammation in the atopic dermatitis model. However, we are interested in understanding what role it played in driving host immune responses. To perform this study, we used a mutant Staph aureus strain that's deficient in 10 extracellular proteases that was previously found to be important for driving skin inflammation. We combined this with our IL-17A/F reporter strain again. And what we found is, as before, in the parent Staph aureus strain, there's a significant increase in the IL-17A and F expression. What was interesting is that in the mutant protease strain, there was a significant reduction in both IL-17A and F expression in the skin, suggesting that Staph aureus proteases were important for driving IL-17 responses. Furthermore, we took the immune cells out of the skin and looked at eosinophil IL-17A and F producing cells. And what we found is that the protease deficient strain, there was a significant reduction in the amount of IL-17A producing eosinophils and a similar trend in reductions of IL-17F producing eosinophils, suggesting that Staph aureus proteases promoted IL-17 responses that were promoted by eosinophils. Psoriasis is another inflammatory skin condition that we were interested in studying. Psoriasis affects 2 to 3% of the population, and although its etiology is not completely known, it involves environmental factors such as stress, microorganisms, drugs, and trauma, in addition to disease-associated genes such as IL-12, IL-4, TNF, and IL-23. Stress cells are thought to release self-DNA and LL37, which complex to activate plasmacytoid dendritic cells and initiate an inflammatory cascade. As disease progresses, there is an increase in IL-17A responses that promote neutrophilic responses, and what our group is interested in understanding is what role neutrophils are having in this response. It's been previously shown that the antimicrobial peptide LL37 and extracellular RNA promote psoriasis inflammation. However, we were interested in understanding what was the source of LL37 and RNA in psoriasis inflammation. Our collaborators found that looking at human psoriasis skin samples, that neutrophils were associated with the LL37 and RNA in the skin. To understand what role this had on neutrophils, our collaborators incubated neutrophils from humans with RNA and LL37. And it was only in this combination of RNA and LL37 that neutrophils reacted by promoting the release of their intracellular content in a process known as neutrophil extracellular traps, which are known to promote skin inflammation in psoriasis. Therefore, what we did next is we used a mouse strain called Lysem EGFP, which allows us to visualize neutrophils in the skin. And what we found is that when we injected RNA and LL37 into the skin, we see a significant increase in the amount of neutrophils that migrate into the skin over time. And this also correlated with an increase in skin inflammation as measured by ear thickness compared to vehicle controls. Next, we were interested in how this neutrophil influx mediated by RNA LL37 injection was promoting skin inflammation. And when we looked at histology, by measuring CYT-H3 and NPO, we found that there is an increase in CYT-H3 and NPO overlap in the RNA LL37 injected skin. And CYT-H3 and NPO are markers of net formation and neutrophils respectively, suggesting that when RNA LL37 is injected into the skin, it promotes neutrophil migration, but also promotes the induction of net formation in the skin, which causes a feed-forward inflammatory loop and promotes psoriasis-like inflammation. In conclusion, we found that in our infection models, using in vivo imaging, we're able to test the efficacy of new therapeutics, such as antibiotics, also testing various dosing mechanisms to understand which may be beneficial, and lastly, different modalities, such as antibiotic-coated implants, to see what effect they have on infections. Additionally, we were able to understand the immune mechanisms in host defense against Staph aureus infections. This included understanding host defense responses and also the mechanisms that promote clearance of bacteria by new therapies such as vaccines. Lastly, we were able to better understand the immune mechanisms that drive inflammation in disorders such as atopic dermatitis and psoriasis, including those driven by the host, such as understanding that eosinophils were drivers of IL-17 inflammation, and also what the pathogens were doing, such as proteases that are produced by Staph aureus in driving atopic dermatitis like skin inflammation. And with this, we hope that we can use these new understandings into developing novel therapeutics against skin infections and aberrant skin inflammation. With that, I would like to thank the current and former members of my lab, Integrated Biotherapeutics, the University of Tübingen, our collaborators at Johns Hopkins Dermatology and the School of Medicine, and various other universities including Mayo Clinic, University of Colorado, NCI, and the TB Alliance. I also want to thank my funding sources and PerkinElmer for inviting me here today, and the audience, of course, for your attention. Thank you. Please email me with any questions that you may have or connect with me on Twitter. And please stay tuned for the next talk by Dr. Stephen Baker. Thank you again.
Good morning, good afternoon, everybody from wherever you're listening from. So it's over to me. My name's Stephen Baker. I'm a microbiologist at the University of Cambridge. I'm going to tell you a little bit about what we've been doing with respect to some work on our AMR, and also how that kind of integrates with some of the imaging work we've been developing with PerkinElmer. So just to change gear a little bit, it's worth kind of refreshing what is the problem with AMR and why is this so important. I was fortunate enough to spend 12 years living and working in Southeast Asia, and there is a circle there focused around Southeast Asia, which represents approximately a 6-hour flight radius from one side to the other. In that circle, and it's pretty movable around that part of the world, it encompasses approximately, I haven't counted them, but a third of the world's human population and almost half of the world's animal diversity. So it's no real surprise that new things emerge from this part of the world on a fairly rapid basis. Combined then with kind of animal husbandry, and people believe, and we can debate this at another time, about the One Health prospect of how circulation in antimicrobials in humans impact in animals, impacts on humans. But also then in the bottom right-hand side there, those are ciprofloxacin that were bought within a mile from where I lived, from about 10 different pharmacy shops, all for less than $1 with no kind of prescription or anything needed. So access is a problem, and it impacts then on generating drug-resistant organisms. At the top right there is an organism. That's my organism. It's a Staphylococcus that was isolated from me, and it highlights the importance of how AMR works. It was resistant to multiple different antibiotics that I was taking, and in the end, it was only susceptible to one that was an oral antimicrobial, and I was about 3 days away from having to go to hospital and have intravenous antimicrobials. So I'm going to try and show an animation if it works. Oh, there we go. Sorry, next slide. So this is AMR emerges because of this. This is the way antibiotics work. They're non-selective, so they work like an atomic bomb. If you take an antimicrobial, they will attack anything that they recognize as a microorganism, whether they are susceptible or resistant. So even if organisms are resistant, they will still have an interaction with that particular drug. Therefore, then we generate antimicrobial resistance because of this broad reaction of way these broad chemicals interact with particular microorganisms. Now we'll play this, and this is an example of why this is so important. So this is then at the bottom a diagram of the emergence of fluoroquinolone-resistant Shigella sonnei. In blue dots, we have fluoroquinolone-susceptible Shigella sonnei over a timeline, and in red dots, we have the emergence of fluoroquinolone-resistant Shigella sonnei. And on the right-hand side, we have a phylogenetic tree of the genome sequences of these organisms, and then we have on the left, we have a map. And as you can see, then suddenly we get the emergence of fluoroquinolone-resistant Shigella sonnei. in about 2009, it forms this clonal expansion and it radiates internationally within the space of about 3 years. It's really tremendous speed the way this becomes transmitted from one place to another. And it forms subclones. So this is an outbreak from that original outbreak in Asia, in Europe. And this is one where I was at the time in Vietnam. But actually, the vast majority of these actually originated from South Asia, and particularly from the emergence from India. So Asia is a big problem, and the way we use antimicrobials impacts on the way they then respond to different therapeutic targets. And this is a problem, and I'm happy for you, if you would like to go and read some of my articles that we've written about this. But this also highlights the data really nicely. This is information from blood culture infections from Nepal over a 20-year period, perhaps. I think it is, I can't quite see, maybe 15 years. So looking at the proportion of organisms that were non-MDR, so non-multidrug resistant, resistant to 3 antimicrobial classes or more, multidrug resistant 3 antimicrobial classes or more, or non-MDR, so less than that or susceptible. And you can clearly see with the different groups of organisms, so gram-negative bacteria, gram-positive bacteria, and others over that period of time, they all increased significantly during that period. And then, as you can see, then there's a period here at the beginning over 10 years where non-MDR organisms were more prevalent in blood culture infections than MDR organisms, and they track each other for a while. And then since about 2009, there's been this transition, and now this has continued, and now almost all organisms isolated from blood culture infections in this part of the world are now multi-drug resistant. And we've done a lot of work on this, really kind of defining the problem and trying to understand a little bit of how it emerges. This has been focused really on multiple different organisms, but Salmonella typhi is one of our key organisms that we work on in the lab. It causes typhoid fever. It's vital really for treatment of typhoid. There's a predicted 20% mortality rate in the pre-antimicrobial era, and we've looked at this over this period of time. Lucky enough to work in a group again in Nepal through a period where we're testing multiple different drugs for the treatment of typhoid, and you can see that the increases of minimum inhibitory concentration of ciprofloxacin, or other fluoroquinolones, compared with flea clearance time. So this is then shows how important this is. That if you have a drug-resistant organism, or the higher the MIC to that organism against gatifloxacin and ofloxacin, the longer it takes for you to clear the infection, and the more likely you are to have more severe typhoid and require rescue therapy. One of our studies on the left-hand side here, during one of the trials, we identified the emergence of a new subclone which actually acquired resistance or hopped into trial participants during the study and was associated with treatment failure, and actually showing that the organism that you were infected with and the genotype of that organism could actually predict how you were going to have your outcome from the drug. And then also then moving into sub-Saharan Africa. So these organisms have originated in Asia, but then radiated across the Middle East and are then causing infections now in Eastern Africa as well. So showing that this is a regional problem, and then it becomes a global problem, and it really does impact on disease outcome. Just to take this on one step further, so if we look at the likes of ciprofloxacin resistance, which is quite common now in almost all gram-negatives, we do have many alternatives still, or some alternatives. But this then highlights how this is a kind of chase that we're never really going to win. So this is XDR typhoid. There's a phylogenetic tree there on the left-hand side of genome sequences, a whole range of different Salmonella typhi. There's a big grey block in the middle that says Pakistan XDR. So those organisms are resistant to all but one oral antimicrobial, which is azithromycin. And this is a problem, and actually it's been important for the introduction of typhoid immunization in the region, because the WHO granted the introduction of the vaccine on the basis to stop this outbreak of XDR typhoid. So these remain susceptible to an alternative drug called azithromycin. But if you look here, we have Bangladesh, their ACRB, and in Pakistan ACRB. These are single mutations in an efflux pump, which render those isolates there resistant to azithromycin. So it hasn't yet appeared in the XDR strains, but actually what we're seeing then is now the emergence of resistance to azithromycin all the way across the Indian subcontinent. So India, Pakistan, Bangladesh, Nepal, and we are essentially one mutation away from having typhoid that is untreatable with all currently available oral antimicrobials, having a big impact then on the likelihood that people would need to be admitted for typhoid and treated with alternative intravenous antimicrobials, or not receiving any therapeutics in the community. And arguably, I would say that one of the organisms that I'm most concerned about would be Klebsiella pneumoniae. So Klebsiella pneumoniae is another gram-negative bacteria that colonizes the gut commonly, but it's associated with a whole range of different infections in humans. It can cause pneumonia, but often it can get into the bloodstream from urinary tract infections or from colonization of the gut and cause fairly unpleasant bacteremia. It can get lodged in the liver and cause unpleasant liver abscesses and other things. So what we find is there are hypervirulent strains that cause this invasive disease. These are not traditionally resistant to antimicrobials. They're difficult to treat because of the way they get lodged systemically, but actually they're not traditionally resistant to antimicrobials. And you can see that from the plot on the left-hand side. So IUC is a locus associated with this hypervirulent phenotype, IUC plus on the right-hand side there, and they're generally less resistant to different antimicrobials. However, you can see two groups there saying ESBL and carbapenem. So those are coming from India, as highlighted by orange, and those are organisms that are emerging in India that are hypervirulent, so contain the genes associated with hypervirulence, associated bloodstream infections that are also resistant to third-generation cephalosporins and also then carbapenems. And how is this happening? Well, it's not the emergence of an individual clone. It's observed in many different sublineages. So we see MDR organisms that are acquiring the virulence plasmids, but also alternatively hypervirulent clones acquiring the MDR plasmids. So this becomes a bigger problem of the way these different virulence genes and MDR plasmids can hop around between different Klebsiella, but also between different members of the Enterobacteriaceae. So that's kind of where we are. And that's kind of been my, sort of snapshot of part of my career for the last, I guess, 5 to 10 years, if you like. So now we're really in a position where we need to start thinking about what we can do about it. And it's a major challenge. And I think that as with other big major challenges, such as climate change, there are lots of things we can do, of which one of them is potentially nothing. That we can just sit and see what happens and see then how this pans out with the view of leaving it in the lap of the gods. The other thing that we're very good at as scientists or microbiologists is collecting data, growing organisms, isolating them, doing genome sequencing, as I've just highlighted. And that also then helps us understand the problem, but doesn't overall tackle the problem. It defines the size and the shape and the magnitude of the issue, but perhaps doesn't get where we want to go with a view of coming up with alternatives. So what do we need? Well, we need new drugs. We need better diagnostics to understand how we use the drugs that we've got better. We need new vaccines. We need other alternatives therapeutics, such as monoclonal antibodies. We can think about how we repurpose existing drugs by examining how they interact with resistant organisms. We can think about alternative therapies, such as bacteriophage and other things. We can look at reversion, so we can think about how we block particular drug susceptibility pathways and see whether we can revert them. But also we can then come up with more kind of conventional kind of clinical interventions of how we reduce or manage or introduce antimicrobial stewardship programs. But ultimately, we want to do all of these, and I think within the group, we don't try and tackle everything, but we're thinking about lots of different aspects of these things, and we use lots of different techniques to try and add or generate data in these particular areas. And more, and we're keen then to think about how we could expand this, and other groups are expanding what we can do in this space to really think about how we can tackle it. But actually what we really need is new approaches that we can use to study and create new interventions. And moving on really from the more descriptive microbiology aspects of what we've done in the past. So one of the things then we've been working with PerkinElmer with and ourselves is then using this piece of equipment, which is an Opera Phoenix. It's really then generated for studying mammalian cells more frequently, but we've used it to then study bacterial populations, to study them in great detail. So it's computer-assisted automated microscope. We can run it, and we're running it in 96 and 384 wells. It's really high throughput. You can run 14 plates per stack. And just to show, it's like essentially for imaging, it's like shooting fish in a barrel, because we can look at 200 cells per well and 80 images per bacterial cells, which really gives us an enormous amount of information. So then it ceases to become really an imaging problem or experimental problem, but really a mathematical problem. And then we have different things that we can do about how we use the assay, about how we look at the image, but also then how we use that information and think about how we can move that into using different things in machine learning, artificial intelligence, and other stuff. So we're really just getting going with this. We've had it for a little bit of time. We've got a couple of papers that have come out, but actually we're really starting to get to grips with how we can use this to study bacterial populations, particularly with respect to AMR. And I'll give you a couple of examples now. So this is kind of how it works. And this is one frame of some bacteria I've been working with. I think this is a Klebsiella or E. coli. We can fix it with different stains, as you would do conventionally with confocal microscope, and therefore we can overlay them and do nice things, and then we can merge them and then really look at different binding patterns of different things we can stain on the bacteria. We use the software that comes with the equipment to hone in on particular individual bacterial cells and bacterial cell population. We define what we're interested in looking at ourselves. So we then look at how these bacterial cells are structured, what we're interested in examining from them. We can infer what we want to look at, what we don't want to look at, so we can count live dead bacteria and do lots of other nice things with it. And then we get to process the image data the other side. And this is kind of how it works then. So input the image, we'd filter the image, look at the intensity of the organisms. We can select the populations that we really want to, remove the borders around them, then do the calculation and then define the bacteria. And that then produces a number of different variables that we can then work on and put through various pathways that we're developing in the laboratory. Now, you may ask me about these later. I'm not a mathematician, so we're very much dependent on working with mathematicians to interpret some of this information. But it gives us an output of anything up to 130 different parameters per organism. And traditionally, we're working with about 60 to 80 different parameters per bacterial cell in one plane. So it gives us fantastic resolution. And then this is kind of how it looks. So a little bit more about how we generate that information using different stains with the overlays and seeing how they're bound with different proportions of the bacteria being bound to different things. Great for looking at how antimicrobials may interact with the cell surface or how they may impact on cellular morphology, or also we've been doing some work looking at antibody staining. Okay, so let's look at this. And this is one of the first things that we did really, was to look at salmonella exposed to different antimicrobials. So we took a relatively low concentration of these different drugs and then looked at how then these organisms then reacted to these particular different antimicrobials using sub-MIC concentrations. And we can clearly see then that we can generate different cellular morphologies by exposing these to different chemicals, which teaches us a little bit about how these organisms are interacting with these drugs and potentially how then resistance may emerge by changing the morphology of the bacteria. And there's a principal components analysis on the right hand side there, looking at the different variables, pulling out about how they behave and how they may group together. So taking this on a little bit further, we were particularly interested in ciprofloxacin because it's a really commonly used antimicrobial, but also then we hypothesize that it has varying impacts on cellular morphology and on the way the organism behaves and responds to different antimicrobials. We think that actually it stimulates different pathways within the cell, which may trigger resistance to other antimicrobials. So we did actually a comparatively simple experiment where we took 2 clinical isolates, a susceptible and resistant isolate, and 2 laboratory isolates, susceptible and resistant against ciprofloxacin, and exposed them to 4 different concentrations over a 24-hour period, and then imaged them every 2 hours for up to 24 hours. And those are the images you can see there on the left-hand side that are taken from the opera, and we can quite clearly see that there's some nice kind of cellular elongation as you expose them to different concentrations of the drug over a period of time. So then we grouped this. So then what we did is then took that opera information, we took the 80-something parameters from those organisms and then put them into some mathematical algorithms to work out how they group together. And they group together quite nicely. So what it shows then, there's an association here with the morphology about how the bacteria behave, regardless of the bacterial background, with their interaction with the concentration of the drug. So the 0 and the 1 times concentration MSE are on the left-hand side, and the 2 and the 4 times are on the right-hand side. So as you increase the concentration of the drug, that really has an impact on how the bacteria respond and change their morphological characteristics. Remembering that then this is the multiple different parameters that we take off the opera. But also then we can look at this in a little bit more detail, if you like, with how this affects with time. And this is probably less obvious, but if you kind of squint and you look on the right-hand side, you can see more yellow and green, and those group together, which suggests actually that independent of genotype of the organism, the longer you expose the bacteria to the drug, the more they group together morphologically using these characteristics. So why is this important? Why is this remotely relevant? So if we then look at this again and look at the distance ratio, i.e. the way these bacteria behave through time, we can see a substantial drop off there at the 22-hour time point. That means they all kind of behave similarly at that point. And you can see that actually at those concentrations of the drug, they all drop, including the 0 times MIC. And this is really quite interesting because what it shows then is that these bacteria behave very similarly at a 22-hour time point, whether you expose them to an antimicrobial or not. And then if we take that information and then run them together and do a cluster analysis, we can group quite nicely the susceptible and the resistant organisms from that particular experiment together without ever exposing them to the antimicrobial. So actually we can then distinguish the difference between susceptible and resistant organisms purely on those characteristics we take from the microscope without ever exposing them to the drug. And this is again what it looks like. So we can predict then with a collection of different bacteria how this behaves. And we've done it with a number of, followed up from this using the machine learning, looking at these different algorithms of what they are, of taking those key parameters, and then we can then define, using the information generated from only 4 strains, but putting it back onto a larger collection, we can predict whether these bacteria are susceptible or resistant to a particular antimicrobial without ever exposing to that antimicrobial. And we can do this at about 92% sensitivity on detecting resistant compared to susceptible strains. So some improvement required, but what it shows is that the microscope can actually detect subtle morphological variation in the bacterial cell that we can't see with the naked eye that predicts how it's going to interact with an antimicrobial. Now, this is really interesting on the basis of then thinking about how a specific mutation, in this case, a single base pair mutation in the DNA gyrase gene, can then impact somewhat on the morphology of the bacterial cell, which demonstrates how it's going to interact with a specific drug. And this means then we can really think about how we expand this into other areas of machine learning and generating more data, not only to infer how bacteria may be resistant or susceptible on the basis of diagnostic testing, for example, but also to understand more mechanistically how these bacteria respond to these drugs and how drug resistance is generated. The last thing, or the next thing that I want to talk about before I finish, is thinking about Klebsiella and the generation of Klebsiella vaccine. This is enormously complicated. And there have been previous attempts using a whole host of different things and methods. And we've looked at trying to generate some of this ourselves using outer membrane vesicles, but actually getting then a cross-reactive response to them is really, really complicated. And what we've done then is get together a large collection, 175 isolates that we've characterized genetically and performed a load of different morphological testing, both in the laboratory, but also then through the opera, to really characterize this kind of complex species to understand how it may behave with respect to immunization. And what we've done then is taken 5 organisms that are representative, we think, largely of what's going on clinically with Klebsiella, of key sequence types, key capsule types, and key antigen types. We've purified outer membrane vesicles from them, which are kind of blebs off the bacterial surface, and they create a cross-section of the outer membrane, and then immunized mice with them, and those mice have generated then an antibody response. And the question really is then, if we then want to generate a Klebsiella vaccine that induces antibody-mediated immunity, what does that look like against a whole host of different Klebsiella pneumoniae? So again, going back to the opera, we take our bacteria, we take our polyclonal antibody, we then fix our different bacteria, in this case 175 different Klebsiella, and then we analyze the data to really infer how those different polyclonal sera from our immunize bind to a whole host of different Klebsiella. And this is then kind of what the data looks like initially, looking at very limited kind of heterotypic binding, but then high homotypic binding. So actually what we find is that, unsurprisingly, you generate a really strong antibody response against the organism you've immunized from the OMV against, but actually from others you get less cross-reactivity. You get some, but actually it's not as good as immunizing with the host strain. Unless you look at here, serum number 4, where we get a really good cross-reactive response to a whole host of different Klebsiella. So actually not all OMV purifications from organisms are the same, and they do then detect different things that may be more or less cross-reactive against a whole host of different Klebsiella, which we actually didn't know before. And this is then how we're then taking for training a model then of how this may work. And this is something that's ongoing work at the moment, and we categorize these into different things. So here on the left-hand side, we can see strong antibody binding against a particular organism with a particular serum. We see mixed antibody binding, where we see a combination of organisms that are bound and unbound, again, highlighting how important thinking about bacterial populations are in one plane. These are all the same organism growing up on the same day, and they behave differently with an individual plane of experimentation. Weak antibody binding with a different serum, and then also then heterogeneous antibody binding, where you get these clumps and clusters together. And what do these things mean, and which ones are important for immunization? Actually, we don't actually know, but we think that actually getting the most antibody bound to the bacteria is likely to lead to some degree of a functional response. And again, just looking at how this works, some strong binding with another combination of an organism, some mixed binding again, where we get this patchy pattern of some organisms being bound and others not, and then also then weak binding, where we get bacteria that don't show any particular prevalence for binding the antibody from the polyclonal sera. So how does this then look? So these are our 6 sera. These are 175 different organisms looking at the binding patterns generated on the opera phoenix. And when we can clearly see then, as I alluded to before, a proportion of binding across the bottom, that our serum number 4, so from our OMV number 4, we can clearly show the most kind of cross-reactive binding over 50%, or the median is over 50% of those organisms showing strong binding within an experiment. And others drop off a lot less. So now we're working on characterizing what they're binding to, the composition of that OMV, to try and work out what we're triggering an antibody response to, and how that binds, and what those potentially conserved proteins or polysaccharides are between those organisms, to see what we're actually targeting the antibody against. So making sense of the data becomes quite tricky. And there's a lot. So we're working with, as I said, we're working with the mathematicians here at the University of Cambridge to really understand how this then works, looking at the proportion of binding organisms. So we use particular cutoffs. So here, a threshold greater than 600, and a mean intensity of the DAPI stain to show that we're getting organisms in the plane of field, but also the amount of bacteria getting bound. And we've then done this across all of our collection of 175, and now we're trying to work out what that means for generating cross-reactive antibody response, which may induce some degree of protection against Klebsiella pneumoniae. So that really is then a snapshot of where we are. I was hoping maybe to have more conclusive information, but we're just really getting going of understanding how we can use this better. But we know then this is a major health challenge for which we need new approaches. We use a whole range of different methods, of which imaging has become one of them really in the last 18 months. And actually allows us to see the unseeable at a scale that's really unprecedented. So it allows us to generate data really that we're just getting our head around how we can actually handle with the view of what we're trying to assess. But it has really, and we're using it for a range of different applications. So looking at resistance mechanisms, studying antibody binding patterns, and looking at cellular localization. We're also doing some work looking at functionality. So we're trying to set up assays for serum bactericidal activity, but also opsophagocytosis. Looking at blocking virulence factors in multicellular systems. So we have some organoid models, and we're looking at whether we can block particular surface proteins interacting with those. And also then now we're just starting a project looking at killing and predicting killing using synergy of different antimicrobial combinations. And it's been a substantial effort and investment to get to this stage, but I think we're finding making new discoveries fairly regularly. It's just working out how best to use the technology. And actually what I was hoping to talk to you about, but if I'm invited back, I'll talk to you next time, but actually using the opera, we've actually found potentially the first ever monoclonal antibody for AMR bacteria. So we have a really exciting discovery using a different organism, using the opera phoenix to screen a whole collection of bacteria to identify a particular monoclonal antibody that we think can show some therapeutic potential, but how we prevent or treat infections with such organisms. And hopefully, again, if I'm invited back, I'll tell you next time. And many thanks. So the group here, my overseas collaborators, my previous organizations, some links in with industry, and also then funding organizations that have kept us going for the last few years. And I believe there may be some questions, and I'm happy to answer them if I can. Thank you very much.
And I want to thank you both, Nathan and Stephen, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So it looks like we already have some great questions coming in. Stephen, why do most AMR organisms originate in Asia?
Yeah, I alluded to it a little bit in the talk. I think ultimately we don't know the exact reason for it, but probably it's just overwhelming interaction of these particular chemicals with humans and animals in that location. So the more biomass we have in people and bacteria in one location, the more they're exposed to drugs, the more likely they are to emerge. There's other reasons such as economics and other things in healthcare, but actually probably it's a numbers game whereby the more people we have in one location, the more they're exposed to drugs, the more resistance we have emerging.
And thank you so much. And is a cross-protective Klebsiella vaccine a true prospect?
Well, this is something that's been debated for some time. Our data suggests that probably not. So then it's defining that really individual protective antigen that we can then put into a system that allows us to deliver it. What we may be able to get to is a platform that allows us to deliver multiple antigens in one go, but it's a mechanism then of using systems like I've just defined to actually work out what those antigens are and how then we combine them in an individual delivery system to show them to the immune system. So at the moment, probably not. But as we learn more about how we can generate protection using the type of things that I've just showed you about, it may actually be possible in the future.
Thank you so much. And I want to thank our audience for these great questions. We have one more coming in. Can imaging be linked with any other data to infer AMR profiles, Stephen?
Yeah. So again, we've realized that having, not that to generate more and more data for no purpose, but having more data is always nice, and mathematicians like having more information to work with. So we've actually been doing some mass spectrometry as well. So we take the organisms, we're just analyzing these data now, which has taken a long time. So we've looked at these organisms, we've exposed them to different antimicrobials and different antimicrobial combinations. And at the same time, we've imaged them, but also exposed them to mass spectrometry. And that then allows us to look at not only then the cellular morphology of how that may impact on them to predict drug resistance, but also what's being expressed and what's being shown on the bacterial surface as well. And we think that combining that information really generates a kind of detailed insight into how the bacteria are responding to the drug and how we can predict antimicrobial resistance profiles.
Stephen, thank you so much for joining us for this Q&A portion. Do you have any final comments for our audience before we go?
No, just thanks for listening.
Well, I want to thank you and Nathan for your time today and for your important research. And I'd also like to thank LabRoots and our sponsor, Perkin Elmer, for underwriting today's educational webcast. Before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.